Okay, so welcome once more to the webinar Promoter Analysis with Genomatix MatInspector. I will give you an overview of the foundational program for actually doing transcription factor binding site analysis with Genomatix software, MatInspector. The MatInspector is a program that you can access via a web browser if you go to www.genomatix.de and log in. I already logged in here on this system. And if you proceed to the overview page of the Genomatix software suite, you will find the MatInspector here in this navigation bar under gene regulation first entry actually. MatInspector uses the information that we compiled in the MatBase database, including transcription factor binding sites, transcription factor binding site families, and a number of other things that give you additional information about matches in a promoter, actually in any sequence that you can input here. I will start with showing you a straightforward example how you can run an analysis with MatInspector. You actually have different options how you can enter sequences or select sequences with MatInspector. The first example that I will show you is how to search binding sites actually in a promoter by just entering a gene name, selecting the promoter, and then starting MatInspector. Alternative ways how you can input sequences are shown down here. You can upload sequences from any source actually in a FASTA file or in plain text. You can enter an accession number for let's say an RNA sequence, and you can then let the system also check whether your sequence can be mapped to the genome database and find the corresponding promoters for the analysis. So different ways how you can enter sequences, and we'll just start with entering a gene name. I will enter the SOX3 factor, which is a short for suppressor of cytokine signaling 3. This is a regulatory protein which is induced by cytokines and downregulates or suppresses cytokine signaling by inhibiting the JAK-STAT pathway. And after I entered text here, I get a drop-down list where I can select from which organism I actually want to choose my SOX3 genes. You see the list down here, and I just take the first one here from human. We will search for transcription factor binding sites which are in our database as weight matrices, and just continue here. In the first step, after we selected the gene, we now can select a promoter or the promoters. Here in this case, we have only one promoter actually. And you see that this is the promoter is listed here with an internal identifier and with this position information, and the length of the promoter is also shown here. And it also shows you or tells you which transcripts we have in our database which are regulated or controlled by this promoter. The promoter length is fixed length, which is generally 500 base pairs upstream of the first transcription start site that we find here among the controlled transcripts, to 100 base pairs downstream of the last transcription or most downstream transcription start site. You see that the first transcription start site, the most upstream one, is at position 501. here in this promoter. This belongs to a transcript that comes from RefSec, which you can see at the hand of the accession number here, which starts with NM. So a coding transcript from RefSec. We also have other coding transcripts here. This one comes from GenBank. So we include different sources of transcripts in our database. And this one here comes from Ensemble. And there's also a non-coding transcript here also from GenBank. This information about the transcription start sites can also be used in the next on the next page, in the next step, if you want to have the output of your transcription factor binding sites relative to the transcription start site, so that you get, for example, negative values for binding sites that are upstream of the TSS and positive values for the binding sites that are downstream of the TSS. For those of you who also have licensed the Genes and Genomes package, you have an additional information available. If you click on such a link here, starting with a GXT, this is short for Genomatix transcript and an internal identifier. And behind this link, you can, if you have the license, you can get some information about the expression of the corresponding transcript based on cage tags that we compiled from the Phantom 5 project, so that you see the number of cage tags that have actually been mapped to the genome at the start of the corresponding transcript in different tissues, indicating the expression strength. So this is based on messenger RNA sequences. And in such cases where you have not only one promoter but several promoters, and you are interested in, okay, what is the most dominating promoter in a specific tissue, you can check this information by using the links here to the corresponding transcripts, only if you have Genes and Genomes licensed. Okay, so starting or continuing here, the promoter is already selected. We can just continue in the next page, where we set the number of parameters for our search. And the most important ones are what you see here, the matrix group, so a group of the binding site definitions which are automatically set to the correct group if you start with the gene search. So in this case, the program already knows we come from human, so it selects the vertebrate matrix group. If we came from a plant, it would select the plant group. What is always included are the general core promoter elements, which include, for example, a Tata box or initiate region or motif 10 regions, which are part of the general core promoters. By default, we will use similarity thresholds that are, well, set by the system. I will come back later to that in case you want to change that, or in which cases it might be useful to change them. We will show additional lines of evidence. This is an important output that you get, and which is meant to help you to prioritize your matches according to additional, well, information that we have pertaining to whether a specific binding site could actually be a functional binding site. By default, all results will be shown, both the matches and the statistics. Under certain conditions, you might only want to see the statistics of your match numbers. I will have an example for that as well. I will also select or change one setting here, the offset for the match positions. I mentioned the transcription start sites. Now, if I want to show all the matches relative to the first transcription start site at position 501 in one of the transcripts. In the RefSec transcript, actually, I enter minus 501 in here. Extra output will not change anything here, but apart from the graphics that can also be shown, you can also show the matches aligned with the sequence, and you can allow the extraction of the matches or the extraction of the sequences actually at these positions. Okay, so starting this, we can just submit the search. It will take a little until everything is analyzed. Specifically, the additional lines of evidence need a little to be retrieved from the database, but it should not take more than maybe half a minute or so until we get something. Oh, it's already here. Okay, so in our result, we now get a list of the matches. In total, we get 274 matches, which is pretty typical for a vertebrate promoter. So 200 to 300 binding site matches in 600 base pairs. If you have a promoter that is longer for some reasons, because, for example, because it has a number of different associated transcripts with different transcription start sites, then you might also get more than 300 matches. Okay, what do we get? You have actually different tabs here that you can select as part of the output. First of all, here the match details tab. We have a list of the transcription factor binding site matches. The list is sorted by the position on the promoter. So it starts here at position minus 492, which is the anchor position of the binding site. Anchor meaning it's the central position of the actual binding site. This binding site here goes from position 497 to 487, so on the upstream of the transcription start site. So we have about 10 nucleotides size, 10 base pair size. It's a MIZ1 binding site. You see the matrix family here, $MIZ1, which is short for MIC interacting zinc finger protein. We group our binding sites into families, which you see here, and the families represent actually groups of transcription factor binding sites which are very similar to one another, which are based on information we have about related transcription factors. For example, stat factors, you could have stat1, stat3, stat5a, stat5b, and their binding sites are very similar to one another. We group them into a family and report only, by default, we report only the family match plus the best matching matrix within each family. And this matrix here is called $MIZ 1.01 in this family. And that's, yeah, you see here the information about the matrix itself. We already talked about the positions. Then you have a strand information, whether it's plus strand or minus strand. You have a value for the matrix similarity. So this is a measure how similar the sequence at that position is actually to the binding site definition that we have in here. You get a green background in all cases by default, meaning that the so-called optimized similarity threshold that we calculate for each binding site definition has been met. The threshold can be, or the matrix similarity that you find here can actually be lower than the threshold if you change the parameter for the minimum similarity that you allow, but by default the optimized matrix similarity must be met so that you'll only get matches with a green background here. The next two columns here show you information about the additional lines of evidence that we have in the database for the corresponding match actually being potentially a functional match. This column here shows you the number of different lines of evidence that we have available, and here we have the evidence actually lined out. I will show you shortly what types of evidence are actually available. And in the last column here, we have the sequence at this genomic position where we have the match with well, in red we find positions where we have highly a match to where we have the highly conserved region within the match actually, and in uppercase letters we have the most conserved region of in the binding site, representing the so-called core of the binding site. Okay, now you can sort this list and you can filter the list. You can sort the list by clicking on one of the headers here. So for example, I want to sort the list by the number of different lines of evidence that I have. I click on the header. Well, clicking repeatedly will change the sort order. So if I click twice, I get the matches with the highest number of lines of evidence here. And here this match here is an SP1F binding site, GC box factor SP1GC. This actually has 5 different kinds of lines of evidence, and this is actually the maximum that you can get for a binding site. So to explain what these kinds of evidence mean, here in a reddish background color, you have information that is based on a expert curation of the available literature. And here in this case, it tells us that the SOX3 gene from which we derived our promoter is actually regulated by a transcription factor that can bind this binding site. The transcription factor here is called SP3. And you have additional links here that show you details about the evidence that gave rise to this entry that we have here. Another type of additional line of evidence, weaker certainly than what we get here, is based on co-citation from automatically extracted data. So this is co-citation within PubMed abstracts, and we find that well, we have co-citation of SOX3 with both these factors SP1 and SP3, which can bind the binding site here. Then in blue, you have information that this match here is actually part of a so-called model match, actually of a few different model matches. Model match meaning we find a pattern of transcription factor binding sites of which this binding site is part of, a pattern which has already been described in the literature as being functional in a specific context. This does not mean that it is functional in this context here in the SOX3 promoter, but the pattern has already been found, maybe in another promoter, to actually be functional. So this pattern constitutes a cis-regulatory module where you have well, interaction between the transcription factors and maybe a additive or even more than additive regulation by transcription factors binding to the binding sites. So in this case, this one here would be a combination of binding sites of SP1F and of EGRF, which is for can actually have a look in here. For the EGR1 binding site binding sites for the EGR1 factor in this. So this would be a information about detailed information about this model where it the binding site actually is part of, where we have the definition of the pattern where the two binding sites are on the same strand and they have a distance, well, they're very close to another distance of 0 to 6 base pairs. And here we have the actual reference where this combination of transcription factor binding sites has been described in actually in this case in different promoters, so the promoters of these genes that you find here as a functional combination. The next one here tells us that the binding site overlaps with a so-called constrained element. So this would be a sequence on the genome which is evolutionary conserved among 29 mammals actually, or from the 29 mammals project. So this tells you we have evolutionary conservation of this region, and maybe this binding site is also evolutionary conserved, which could be an indication that it's actually a functional binding site. And lastly, we have information that this in silico binding site also correlates with an experimental binding site, so with a chip sequencing peak for transcription factor here for SP1. These chip sequencing data come from the ENCODE project, or they come from other sources in the literature which we have also compiled and integrated into our database. The information, if you want to see more detailed information about the co-citation that gives rise to entries like we have here, you can also click on such a link, and this will open the Genomatix MatInspector literature mining tool, where you have the corresponding literature outlined here. In this case, we have a publication from 2012 with the corresponding PubMed link, so that you can also open the PubMed link so that you directly jump to the abstract here. And you also have highlighted terms within the sentences here from the abstract, which you can use to further evaluate the information that this is actually a target, or SOX is actually a target of SP3 in here. Correspondingly, this is also same here with a co-citation link. So in this case, it might be more important to actually check whether the co-citation also means a functional relationship. Here you can rely on that, but here in this case it could also be the case that you only have a co-citation but no information about a functional relationship for the corresponding gene here. See, we have more entries here for different publications, and you can just scan the text that you see here and see whether this actually tells you that there is a functional relationship. Okay, now yeah, we sorted. You can also filter here. The list is filtered automatically while you type. And you can, for example, filter specifically for entries where you have this kind of information that SOX is regulated by some factor that can bind here, just by typing is regulated. It's already enough here. Now we only have those entries left where we have at least this type of information. You see that we have 30 entries. We can show more records here per page. By default, we show only 20. If you go to 50 here, then we can see the whole list here in one go, and you see such diverse information like regulated by SP1F, regulated by stat with this stat binding site here, or regulated by BRAC with this BRAC binding site, etc. Okay, another way how you can filter, if we just remove this once more, we go back to our 274 entries, is, okay, now we can filter for specific binding site matrix. So for example, this SP1F binding site, how many do we have in here all in all? We get 13 SP1F binding sites. And well, some of them with a lot of additional lines of evidence, still sorted by the number, and some of them with a little bit less. And we can directly go from here to the graphical view if you want to see the matches on the promoter. So just click here on the graphical view, and what we see here is, well, all of the transcription factor binding sites shown as, well, boxes, rounded boxes in different colors. You see them here on the plus strand, you see them on the minus strand down here, and you also see here is bent arrows, the transcription start sites of the corresponding transcripts. You see there's more than one arrow because we have different transcripts with slightly different transcription start sites. Here on the right-hand side, you can select specific transcription factor binding site families, and you can also deselect all of them or select all of them in one go. So for example, if I click on deselect here, everything is deselected, nothing's left here, and now I want to see only the SP1F binding sites, which I already have in the match, have filtered in the match detail list. I scroll down a little bit and I find them here, so that I now see where my binding sites actually are. And if you look at these, well, specifically here, there's a cluster, and there's maybe a bit smaller cluster. And if you go back to the match details here, we see that, well, starting here from the third one here to the third, fourth, 5, 6, might want to actually change the order here actually. Yeah, so if I sort it now by the position once more, I find that here from these 4, which correspond to the cluster here, this small cluster, these binding sites overlap with here they both overlap with a constrained element, and they also correlate with the ChIP-seq peak for SP1F. And the next 4 that we see here, they still correlate with the ChIP-seq peak for SP1F. So they might actually all contribute to the regulation by of this promoter by SP1 factors. Okay, you can always also save the graph output here as a PNG graph or as a JPEG graph. One more thing here is the match summary table. In the match summary table, you get an overview of the, well, total number of matches for each of the binding site families. You also get a p-value in here. The match summary table gives you, well, a bit of an information about the overrepresentation information, the p-value actually. So the p-value that we have here is the probability of finding the same or higher number of promoters with at least one match for the binding site in the promoter background. The promoter background is for vertebrate binding sites are all promoters for annotated human, rat, and mouse genes. For plants, it's Arabidopsis mainly, and well, depending on the origin, you have a different background. This p-value is useful mainly if you input several promoters from different genes. So if, for example, a set of promoters from genes that are co-expressed or have related biology, to find out whether potential targets of a specific transcription factor family are overrepresented in the gene set. You can sort the list by the p-value here. If I click here, I see the I get the lowest p-values appear. For one sequence, as we have it here, it tells you only in the end whether the transcription factor binding site is relatively specific, so whether fewer promoters actually have a match, or a more general transcription factor, meaning that the majority of the promoters will have a match. So with a smaller p-value, you have a more specific binding site. With a larger p-value, you have a more general binding site. You should not use it as a threshold to say, okay, everything that is below 0.05, for example, is interesting, and everything that is above 0.05 is not interesting. There are binding sites that you find in 50 or in 70% of the promoters. They are just more general. It does not mean that they have no function in the specific promoter that you are looking at if their p-values are higher than 0.05. Also, the number of matches per promoter does not have any influence of the p-value here, because we do not count the matches. We only count the number of sequences that have at least one match. So here we have, for example, you can filter here. We have 18 binding sites for EGRF and the p-value of 0.14 approximately, and this would stay the same if we had only one binding site for EGRF here in this promoter. Also, the p-value is only correct if the input sequences have approximately the standard promoter length of currently about 600 base pairs. The standard promoter length will actually change to 1100 base pairs in the next major update. Okay, another tab that you have here is the export tab. Here you can export the sequence in or you can export the matches in Excel format. Just show you this here, or in a tab separated format. So if you open matches here with Excel, you now get the filtered matches that you see in the detailed view as a tab separated list or a table here in Excel. Okay, this is basically the basics. A few more filtering examples before I show you a few more options how you can actually input your data in here. Filtering example, we can filter here for model. Sorry. And this will mean we get only those matches where we have also a match to a model, and we can, for example, sort this again by the sorry, need to remove this one here. Okay, now we get all of the models, not only those where SP1F is part. Here this one is, for example, a match of RxRF, RxR heterodimer binding sites, which is part of a model that consists of 3 binding sites, an RxRF binding site, an SP1F binding site, and another RxRF binding site. And you see that here is another one, SP1F binding site, which is also part of this model match, the same here. And this one down here is also a part of this model match. So we find the 3 matches here, which could be functional in this context. In this case, we also find co-citation with a corresponding transcription factor here for RxRF, and again we find certainly also the information that SOX3 is regulated by the SP1F factor, which we already saw. So this is another way how you can filter for promising candidates. Down here we have an EGRF SP1F match. This one, and one that is with, or this one is the other one. It's also part of this EGRF SP1F match. If we filter here in the evidence for ChIP-seq, then we get only those matches where we have the matrix families that have also experimental evidence for binding here in this promoter. You can sort for the matrix family in addition, and now you just get all of the matches where you have an overlap of the transcription factor binding site sorted by the type of transcription factor binding site, an overlap with a corresponding ChIP-seq peak, as you see it here, for example, for the AP2F binding site family. All right, so this was how you can go about the analysis. When you just start with a gene and select the promoter or maybe a few promoters from this gene. Now what can you do if you have not only one gene but maybe promoters of a number of different genes? I show you one example, just a few slides here to give you the information about what the actual background is. Just go back here to the input page of MatInspector. I already uploaded a set of promoters which you find here or see here in this list. You can add sequences from anywhere on your own promoter, on your own computer. And this is called DUX4 targeted promoters induced genes. Background here is just a few slides, 2 slides actually, to show you that this is from a public data set that has to do or studies in a model system a specific type of muscular dystrophy, which is called facioscapulohumeral dystrophy, or in short FSHD. This type of muscular dystrophy is hereditary, and it affects mainly the face and the upper limbs here, so the shoulder blades and the upper arms, and later on also the calves. Onset is usually in early adulthood, and it's a rather common genetic disease of the skeletal muscle with more than 800,000 people affected worldwide. It is kind of a epigenetic disease, you could say. The genetic cause for this disease is an expression of a usually silenced transcription factor retrogene, namely for this DUX4 transcription factor in the skeletal muscle, which is caused by, well, in most cases by a deletion in a macrosatellite repeat close to the end of chromosome 4, which has as an effect that the whole region here is hypomethylated, meaning we have active DNA, and the DUX4 gene is expressed where it should not be expressed, and generates all kinds of mayhem then in the cells. And the data that I used here was from this publication here in 2016, which used a model system, namely immortalized human myoblasts with an inducible DAX4 transgene. And what we are looking at here, if we look at the promoters, which are all in all 44 sequences, these are the promoters of those genes that were both upregulated in this model system by DAX4, and also where it was found that the DAX4 actually binds the promoters using chip sequencing data. So the expression data were generated using RNA sequencing data. So here we have the direct targets of this DAX4 gene, which well, we would expect to also have maybe a corresponding binding site in the promoter. And if you use this set of promoters, 44 promoters, you can expect to have several thousands of matches, as you typically have approximately 300 matches per promoter. With 44, you can expect to have about 12,000 matches. There is a limit actually to MatInspector how many matches are displayed. So if you upload a relatively large file with a lot of sequences, you may not get the complete result. So this is a limit that we have in MatInspector. What you can do, however, is you can, if you go one step further here, just to the next page once more, you can let the system only show you the statistics of the match numbers. Will not show you all the results. There is no limit to that. So you can, if you run it then like that, this will just show you the overview page. With a match summary table, and you can once more click here to see to sort the list here by p-value, and you find that the DAX-F binding site family, which is the binding site in our system for DAX4, is at the top here with a very small p-value. So this means we have we actually do have an unexpectedly high number compared to the random background of sequences where we do find a match. We find a match not in all of the sequences here, 44 in total. We find at least one match in 34 of our input sequences, and we also find overrepresentation here of another some other transcription factor binding sites also from the homeobox family. So DAX-F, it's another thing that you can have a look at. If you click on such a link here, then the corresponding page in the mat base will be opened, and you get all types of information about this transcription factor binding site family, including match numbers, and the matrix is here. So this would be the actual binding site definition. You can click here once more, and then you get detailed information about the binding site, where it was defined or described, where we have it from, and also here, for example, the sequence logo of the binding site, where you have the sequence here, and the height of the stacks here represents the conservation of each position within the binding site, plus some more information here. Okay, so that's if you for a statistics output only. If you have a large number of input sequences that you want to analyze in one go. Alternatively, if you still want to stick with a lot of sequences and you are aware of the limit for the number of binding site matches, you can also limit your search to specific binding sites. So it's not necessary that you let the system search for all of the binding sites within each of these groups, but you have the option to continue here with a subset definition from the selected groups, and you can also save these subsets and you can reuse the subsets in later analysis. So if we want to do that and want to look only for the binding sites for the DAX4 in our promoters, we can continue here and can again submit, and then we get an extra page. And on this extra page we get to select what types of families we now actually want to see. Now I just jump to the DAX-F binding site family down here and select this, scroll down to the end. Here I could save this as a subset under a specific name, which I don't do now, and then I can start the search. I will show you that here as well, change thresholds. Sorry, I forgot. We need to see everything certainly. Okay, once more, search for DAX-F. Start the search. So now we should expect to get at least one match in 34 of 44 input sequences. If we don't find any match, the corresponding sequence or the corresponding promoter is just listed at the end of the output that we get out of this here. It will take a little longer because now certainly we have to search through a number of binding sites or a number of sequences in order to find the binding site. Here comes the result, which will then just look like this. We have, again, for each of the sequences, we have our list now containing only the DAX-F matches. And if we scroll down to the end of the list, we also get a list of the promoters that do not have any match for this binding site. Graphical view will include all of the sequences, and you see, for example, here we have a single binding site here. We might have some instances where there isn't anything, so no binding site here in this promoter, and here we have a whole cluster of binding sites. Now, for specific under specific conditions where you can expect or where you know that a specific transcription factor actually binds your promoter and you don't get a match, for example in the sequence that we have down here, you could carefully change the thresholds for actually finding such a binding site. And this should not be done if you don't have any additional line of evidence that there is actually a binding of a specific factor. But if you do know, like as we have here, because we have, well, experimental evidence, you can carefully change or reduce the threshold here for the matrix similarity from optimized to, well, a slightly lower number here. Be careful if you do that. I would not go down far below optimized minus 0.03 because you will certainly increase the chances of just getting random hits, of just getting, well, maybe a few weaker binding sites, but then if you go down to minus 0.07 or something, then it will be completely unspecific. You can, if you change this, for example, to a slightly lower value than the optimized similarity threshold, keep the core similarity, which in this case means at least 3 of our positions within the most conserved region of the binding site need to have a match. Then you get a few more matches, not many more matches. Do the subset selection once more. And these additional matches will then appear instead of with a green background, actually with a red background in the column that shows you the matrix similarity at that position. Result is already coming through. Okay, now here we are, and you see that you now get entries like this, where you have a red background for the matrix similarity. This means this is a suboptimal match, so which is, well, maybe a weaker binding site than the other ones that you see here. Now we get matches not in 34, but in 38 sequences, if I'm not mistaken. So if you just go down to the list here, 6 instead of 10 of the sequences don't have a match for DAXF. Okay, just one more thing here, the last one actually. Alternative to analyzing the sequences that you upload here directly, you can also let the system just search the promoter for a sequence, for a cDNA sequence for you. I already uploaded a file here with a cDNA from the rat for the IL-10 gene actually, and now I can, as I'm not interested in the binding sites of the cDNA itself, but I want to see the promoter, matching promoter, I can just let the system check the sequences against the genome database. Here I should use rat certainly, and then continue here, and the program will map the cDNA to the genome, and I can now select the corresponding promoter here and run an analysis. And yeah, so I'm not going into the details anymore here because we already saw that. Okay, so much for what I wanted to show you today. Okay, questions. I see one question in here. How do I get different transcription start sites? The different transcription start sites, if we go back to the to the output here. So if we go to we have the SOX3. Reason for different transcription start sites are actually different different transcripts that are annotated in this area here, and you can see that it's not part of the MatInspector license, but if you want to see, I can show it here in the Genome browser. So the Genome browser is also part of the Genes and Genomes package that you also can license. And this will open a page where we see the locus, the SOX3 locus. And if we look at the transcripts here, we see, well, we have 4 different transcripts in here. They start here, they are on the minus strand, shown here by the arrow that points to the left-hand side. And if you zoom in a bit more, then you see that the annotation for the transcripts does not start exactly at the same position. That's why we get different transcription start sites, because we have these transcripts from the different sources and their sequences and how they map to the genome here. So we might get slightly different transcription starts. Okay, I don't see any more questions at the moment, so I'd like to, well, thank you for your attention. And if you want to learn about a complementary strategy for promoter analysis using comparative genomics in order to find evolutionary conserved transcription factor binding sites, which is another way how you can select transcription factor binding sites matches for most promising binding sites, watch our next webinar in approximately 1 month on Wednesday, December 7. Then I will talk about gene to promoter for actually retrieving promoters from different genes. I will talk about common transcription factor binding site search. I will talk about dialign TF, which is an alignment program that includes information about conserved transcription factor binding sites, and maybe a few other programs that you can use for comparative promoter analysis. In any case, we'll send you an invitation to this webinar soon. And if you are interested in more, then I'll just register for that webinar as well. I'll be happy to meet you then once more. This leaves me to wish you a nice day and a nice evening, depending on where you are, until we meet again via the webinar series next time. Bye-bye.